So let's take a look at two of the key cycles in cellular energetics. We're going to start with the citric acid cycle, which is a part of cellular respiration. And so this starts with acetyl-CoA, which is derived from glucose, and that combines with oxaloacetate. We lose the CoA, so we combine a 4-carbon molecule with a 2-carbon acetyl group and create a 6-carbon citrate, and that's the citric acid that the cycle is named for. Then that citrate is oxidized. It's going to lose some electrons, and the electrons it loses are passed to NAD plus to create NADH. And as we do that, we're stripping away CO2 molecules from that 6-carbon molecule, and what we're left with is a 4-carbon molecule. And through a number of changes, that 4-carbon molecule, in this step, it's used to synthesize GTP, and that GTP can be used to synthesize ATP. And then again, with another change, we're going to reduce FAD to FADH2, so now it can carry electrons. And then again, another oxidation reduction step. So we have moved the electrons from the carbon-based molecule to these electron-carrying nucleotide derivatives. Also critically important is that we have restored oxaloacetate, so that molecule is ready for more cycles of the citric acid cycle. Now let's take a look at the Calvin cycle. So the Calvin cycle is also referred to as the light-independent reactions of photosynthesis. And in this step, we're going to take 3 RUBP and 3 CO2, and we're tracking 3 molecules at a time here. That's important later. And that's going to synthesize or create 3 unstable intermediate molecules. There's 6 carbons right now, and that's mediated by the enzyme Rubisco. And because those intermediates are unstable, they're going to break into 6 3-carbon PGA molecules. Those 3-carbon molecules are modified using ATP. So you can see that at the beginning, it only has 1 phosphate group. And later on, after we have removed the phosphates from the ATP, we have 2 phosphate groups here. So this would obviously be 6 ATP molecules that we're using to mediate this step. And then we're going to, with another transfer of energy, create 6 G3P. And we're using energy from NADPH this time, so we're moving those electrons to the carbon-based, the 3-carbon molecule. And we're going to get 6 G3P, and 1 of them is going to leave the cycle. And that's why we had to start with 3 CO2s, 3 carbons in, we allow 3 carbons to leave. And the rest of the cycle is devoted to restoring those RUBP molecules. So we have 5 3-carbon molecules left for a total of 15 carbons, and we're going to have 15 carbons in the 3 RUBP. So the energy required to do that comes from ATP, and we restore the RUBP. So we're back to the beginning of the cycle, just like we were with the citric acid cycle. We have to restore the molecule that we started with so that the cycle can continue. So I want you to be able to look at these side by side. So let's start with cellular respiration. So it starts with glycolysis and takes a glucose to a pyruvate, and that pyruvate molecule is going to lose a CO2 and become acetyl-CoA, and then the entire citric acid cycle can run, generating those electron-carrying coenzymes and ATP through substrate-level phosphorylation, and then you go to an electron transport chain, which takes the electrons from NADH and FADH2, and through chemiosmosis produces ATP. To do that, you have to have a final electron acceptor, which is oxygen, and so you generate H2O as the oxygen accepts the electrons at the end. On the flip side, and this literally feels like a flipped reverse mirror image sort of a deal to me, you have the cycle at the end of photosynthesis, whereas it was sort of at the beginning, earlier on in cellular respiration. So you start here with the electron transport chain, which is the light-dependent reactions, and that removes electrons from water, creating oxygen, and uses those electrons passing through the electron transport chain to generate energy for NADPH and ATP. And those electron carrying molecules go to the Calvin cycle so that what you end with is a large biomolecule in the form of G3P, whereas what you started with in cellular respiration was a large biomolecule. So these two processes are beautiful mirror images of each other, and I hope that helps you understand the two cycles separate but also in relation to each other.